Hello, everybody. Thank you for being here today for our webinar session. Our webinar session is under the patronage of the Pharmacogenetic Network, African Network. So let me introduce you to the African Pharmacogenetic Network, which aims to collectively build the capacity for pharmacogenetic research and implementation on the continent of Africa. It was formally launched on the 6th of September 2018 at the University of Cape Town. This network is affiliated to the African Society of Human Genetics, and the membership is open to all interested stakeholders. So today we will have a very nice session with Professor Cortez. Professor Cortez is the head of pharmacology at the Brazilian National Cancer Institute and the coordinator of the Brazilian National Pharmacogenetic Network. He is a pioneer of pharmacogenetic studies in the Brazilian population. His research explores the impact of genetic admixture on the conceptual development and the praxis of pharmacogenetics, genomics. He is a member of the Brazilian Academy of Sciences, a senior investigator of the Brazilian National Research Council, and investigator of the a senior investigator of the Brazilian National Research Council, and a professor of clinical and basic pharmacology at the University do Brasil in Rio de Janeiro. Thank you so much, Professor Cortez, for being here today, and the floor is yours.
Well, thank you very much for the invitation. It's a pleasure to join this seminar series. And I will present you some of the results from our studies on the implications, the pharmacogenomic implications of population admixture in Latin America. So let me start by, let me see, how is that? Okay. Oops. Well, let me start by showing a little bit of the complexity of population in Latin America. So Latin America is formed by 20 countries and 6 territories. The major languages are Spanish and Portuguese. Portuguese is spoken only in Brazil. And in addition, there are several native languages, of course, from the indigenous people, the Native Americans. And on the right of this slide, you see the population in millions of the various countries in the region. The overall population of Latin America is estimated in 625 million. It's a very heterogeneous and admixed population. This admixture involves 3 major ancestral roots: the Native American or Amerindians, the Europeans, and the Sub-Saharan Africans. And these major ancestral roots have been admixing over 5 centuries. And this has led to the very complex population and kaleidoscopic, as I put in the slide, heterogeneity in Latin America, which results in important pharmacogenomic consequences. An important point is that although all the Latin American countries share the same major ancestral roots, the proportion of these roots vary widely across and within Latin American nations. And I hope that the data I'll show you will convince you of this point. So the data I will present is actually obtained from both our Brazilian pharmacogenetic networks. I will show you data for about 1,200 people who self-declare to be white, brown, or black. Sorry, white, brown, or black, which follow the terminology of the Brazilian census. And these 3 categories of the Brazilian census correspond to about 99% of the Brazilian population. The remaining 1% corresponds to Amerindian populations that were decimated over the colonial period, and also some people of Asian extraction, mainly Japanese and Chinese. In addition, I'll use data from the 1000 Genomes project for the Admixed American superpopulation, which comprises 4 groups: Colombians, people of Mexican origin that live in Los Angeles, Peruvians from the city of Lima, the capital of Peru, and Puerto Ricans. So using ancestry informative markers, we can estimate the proportions of ancestry of these 3 major ancestral roots in the various groups. And you can see that the African contribution is larger in Puerto Rican and in Brazil, is relatively smaller in the other countries of the study, while the native population has a less contribution in Puerto Rico and in Brazil, and increases progressively from Colombia to Mexicans to Peruvians. And the European contribution is large in Puerto Rico, in Brazil, and in Colombia, and it decreases progressively as the Mexican ancestry increases in Mexico and in Peru. But these are average proportions of ancestry. If we look at the individual proportions of ancestry, that is, if we have on this slide, in each slide, there are, like here, 93 columns. Each column has 3 sections in green, red, and blue, corresponding to native European and African ancestry. And what is the major point here, as you can see clearly, is that the individual proportions vary largely in the 4 countries, and the average that we presented in the previous slide does not represent the complexity of the population structure in these countries, nor does it represent in Brazil, as you'll see in the next slides. Here, I show data from a previous study from our group in collaboration with a group from Canada and another one from Mexico, in which we studied the proportion of biogeographical ancestry, native, European, and African, in Brazilians, self-declared Black, brown, and white. And you see that as the proportion of European ancestry increases from Black to brown and to white people, as expected, while the African proportions decrease from Black to brown to white individuals, and the native contribution to Brazilians in general, whether self-declared brown, Black, or white, it's relatively small, much smaller than the other 2 groups, the other 2 ancestries. It's about 8 to 10% across Brazil. Again, if you look at the individual ancestries, you have the same pattern shown for the other Latin American nations. That is, that the individual proportions, here we have 300 self-declared white and 300 self-declared Black individuals. And you see that the proportions, the individual proportions, vary widely across these 2 groups. You are going to have white Brazilians with more than 80% African plus native ancestry and less than 10% European ancestry, but nevertheless declared white, self-declared white. The same applies to self-declared Black individuals, that you have self-declared Black individuals that have only 5% estimated African ancestry. So these are the consequences of extensive admixture in Brazil and in other Latin American countries. And of course, in the case of Brazilians, self-declared race or color, as the Brazilian census describes it, depends very much on the phenotypic characteristics, particularly skin color. And as we know, there are a few genes that determine these phenotypes, and consequently, it's not surprising that you have this pattern in admixed populations. But this will have important pharmacogenomic consequences, and I hope to illustrate that in the next slides. So to summarize this first part, I'll take some of the statements I made in an article written almost 20 years ago, and I think prevails today, that racial, ethnic, or color categories do not capture the diversity of Latin American population, that the distribution of pharmacogenetic polymorphisms among Latin American peoples vary across geographical regions and within self-reported color categories. Thus, ascribing pharmacogenetic polymorphisms frequencies to Latin Americans, or using the term that is adopted in North America, Hispanics or Latinos, is inappropriate. It really does not capture the diversity of Latin American populations. Admixture heterogeneity in Latin America must be recognized in the design, the analysis, and the reporting of pharmacogenetic data, and should be dealt with as a continuous, not as a categorical variable. So one way of studying the impact of population diversity on the distribution of pharmacogenetic polymorphisms is to use the fixation index statistics to compare the frequency of polymorphisms in the 5 populations that are covered by this presentation. That is, the 4 populations of the 1,000 genomes admixed American population and Brazilians. That's what we did in this study, and we analyzed about 1,200 polymorphisms that are included in the DMAT chip. And you can see that for the vast majority of these polymorphisms, the degree of genetic divergence or genetic differentiation is small. And for about 100 of them, it is moderate. And in a small group, the divergence is large. This divergence this large divergence and moderate divergence of frequency of genetic differentiation of some pharmacogenetic polymorphisms can be used to estimate the proportions of biogeographical ancestry. That's what we showed in this study here. And you see here, in this axis, we show the proportions of African ancestry in Latin American populations, all of the populations of the 1,000 genomes admixed American group, plus Brazilians. And here is the estimates of ancestry, of African ancestry, European and native ancestry, obtained with the DMAT panel, that is, with 60 pharmacogenetic markers. And you see we have a very high correlation coefficient for African, European, and native ancestry using either 60 markers, pharmacogenetic markers, or using a genome-wide approach. So that is another evidence that pharmacogenetic differentiation is important in Latin America. It even allows you to estimate ancestry. I will now move into examples of these pharmacogenomic implications and take these examples from the CNPq, the Clinical Pharmacogenetic Implementation Consortium guidelines. For those who are not familiar with those guidelines, this consortium is formed by a very large number of researchers and clinicians around the world. And the CNPq consortium has published about 20-some guidelines, 25, I think, in which there are clear dosing recommendations for various drugs based on genetic polymorphisms. And one of these guidelines is for CYP3A5 and the immunosuppressant drugs tacrolimus. And here are the recommendations from this guideline. And you see that there are 3 groups of 3 phenotypes. Individuals are divided into 3 phenotypes: extensive, intermediate, and poor metabolizers. Extensive metabolizers have no polymorphisms, that is, not the functional polymorphisms used to define the phenotypes. The intermediate metabolizers are heterogeneous for one of the 3 polymorphisms that are no polymorphisms, no function polymorphisms, alleles star 3, star 6, and star 7. And poor metabolizers are those that are either homogeneous for these polymorphisms, these variant alleles, or that are compound heterozygotes for 2 of these polymorphisms. And the recommendations based on these diplotypes are classified as strong by the consortium. And here are the recommendations to start with the usual dose, to increase the starting dose 1.5 to 2 times, the same here, whereas those that are poor metabolizers, you initiate therapy with the standard recommendation dose. So if you look at these 3 variant, these 3 SNPs in the Latin American populations, as we have done in a study that actually informed the CNPq guidelines several years ago. If we look at this data, you'll see that we took samples from Portuguese and samples from a study in collaboration with Dr. Albertino Damasceno in Mozambique. And we studied the distribution of these variant alleles, 3, 6, and 7. They are all no alleles, no function alleles. And you see that in Portuguese, an example of Europeans and the major ancestors of the white Brazilians, you see that alleles 6 and 7 are absent, and allele 3 is largely predominant, while the wild type allele is about 5% frequency, minor allele frequency of 5%. And we see here that in Mozambicans, the situation is completely different, in which the wild type allele is the predominant, and we have a large representation of allele 6 and 7, in addition to allele 3. So now we looked at Brazilians, self-declared white, brown, or black, and what we see is that the average proportion of these 3 ancestries varies progressively from white to brown to black, in the sense that allele 3 gets progressively less frequent, whilst alleles 6 and 7 get progressively more frequent, expressing, as we have seen before, the progressive increase in African ancestry from white to blacks and the progressive decrease of European ancestry from white to black individuals. So this distribution is interesting because the studies that had been performed until the publication of our study were largely done in Europeans, and studies in Europeans do not identify individuals who are homozygous for the wild type allele due to the low frequency of the wild type allele, such that studies in Europeans basically can only distinguish between carriers and non-carriers of variant alleles. Whereas in Brazilians, we have sufficient frequency of homozygous, wild-type homozygous individuals to be able to do studies with a gene-dose association, such that in Brazilians, you can identify poor metabolizers, intermediate metabolizers, and normal metabolizers, whereas in Europeans, you wouldn't be able to see the normal metabolizers. And this is significant because the guidelines are based the guidelines for tacrolimus are based on the phenotypes, as we have seen in the previous slides before. And these phenotypes are classified by the PharmGKB database as low risk for the poor metabolizers and high risk for intermediate and normal metabolizers. That is, high risk implying that there are recommendations for changing the initial dose of tacrolimus in the patients. So we observed that clearly in a large cohort of patients of renal transplant recipients, that we saw that the dose required to reach the target tacrolimus concentrations in blood decreased progressively from individuals who are normal metabolizers to intermediate metabolizers and to poor metabolizers, such that the genotype affects the tacrolimus pharmacogenetics in a clear gene dose manner. And we concluded that the recruitment of the admixed cohort broadens the scope of the pharmacogenetic association between CYP3A5 polymorphism and tacrolimus dose requirements, which were previously observed in relatively homogeneous populations. So we extended more recently the study of the distribution of CYP3A5 alleles among Latin American populations to the 1,000 genomes admixed American populations. And here is the distribution of these 3 variant alleles that are the most important for pharmacogenetic dosing recommendations. And we see that there is a very significant difference in the distribution of these polymorphisms across the 5 populations. However, a very high statistic significance does not imply that there is a very large effect. And indeed, if we use the Kramer's v-test to estimate the extent of the effect, we see that the extent of the effect is small despite the very large statistical significance. This is, I think, an important point for pharmacogenetics, because it is not uncommon to see significant differences in frequency of a given allele or a given phenotype across populations, but that does not imply that that there is a corresponding large effect that will have important clinical consequences. So if we now look at the diplotypes and the phenotypes that are inferred from the genotypes at those 3 loci, what we see is that the distribution varies significantly, distribution of phenotypes across the 5 Latin American populations. But again, the magnitude of the effect is small. And if we look at the priority risk, that is, comparing the high-risk individuals that are those with normal metabolizers and intermediate metabolizers versus the low-risk individuals, that is, the poor metabolizers, we have again a very large statistical significance, but a relatively small effect. Nevertheless, despite the small effect, there will be pharmacogenomic consequences, as we'll see in the next slide. But before that, let me tell you of another approach that we can use for the mixed populations, is that instead of comparing nations, what we can do is to take the ensemble of the Latin American population, that is our Brazilian sample, 1,200 individuals, plus the 370 individuals of the 1,000 genomes admixed American populations. And from these almost 1,600 individuals, select those that have predominantly one of the 3 major ancestries. And we have done that and selected the 80 individuals with the highest proportions of native, African, and European ancestry. And you see that now we have populations, sub-cohorts, that are predominantly native, predominantly African, predominantly European. And now we can compare the distribution of the polymorphisms among these 3 groups. And what we see now when we compare the 3 sub-cohorts formed by the 80 individuals, we see now that we have, of course, a very, very large difference in distribution of the polymorphism across these 3 groups. But now the magnitude of the effect assessed by the Kramer's v-test is very large. So if we think about it, that we are here comparing the average with the individual, because when we look at the frequency in a given population, the frequency in Brazilians, it is 10% of a given allele. But if we know that these Brazilians, some of them have one ancestry, predominant one ancestry, that in that ancestry, the frequency is not 10%, is 60%. And they have another ancestry where the frequency is 2%. So the 10% represents very little of the individual proportions. The average ancestry is important, and the average frequency of the variant polymorphism, or the frequency of the various phenotypes, is important from a public health point of view to determine maybe policies as whether we are going to implement and pharmacogenetic tests in the public health system, for example. But when we think about individual approach to pharmacogenetics, this average approach is not going to be adequate for the extremes in the population that are not represented in the average. So when you look then at the three sub-cohorts with predominantly native European and African, what you see now is that the high risk versus low risk individuals, there's a very large difference, and the magnitude of this difference is very large. So that when we take these data, we have to think about, once we decide on implementing a pharmacogenetic test based on the average proportions, or when we interpret data based on average proportions, we are not considering this complexity that occurs in the highly mixed Latin American populations. A consequence of that is that if you calculate the number of individuals that you need to genotype to find one individual at risk of adverse effect, and we calculate these numbers based on the frequency of the high-risk phenotypes, the frequency of adverse effects, and the odd ratios that of the association of high-risk phenotypes against the frequency of adverse effects, then these allow us to estimate the numbers needed to genotype to identify one individual at risk for an adverse effect. We don't see a very large difference. There is a difference, but not a very large difference between the various populations. But if you look at the subcohorts that represent within those populations, those with largely predominant African, European, or native ancestry, you see a very large difference. We need to genotype about 50 or 40 individuals with more than 90% African ancestry to identify one at risk of adverse effects in the case of CYP3A5 polymorphisms, and we need to genotype 560 native individuals to identify one at risk, and Europeans stay in between in this case. This will, of course, vary with different polymorphisms, with different associations with side effects, or different associations with dosing requirements. This approach can be used for various purposes. And depending on the number of individuals you have to genotype, you can calculate how much it costs to identify one individual by applying pharmacogenetic tests. And these are the costs based on in our lab, how much it costs to genotype these three polymorphisms that allow us to define these variables. And you see, as I said, there's not a very large difference among Latin American populations from 930 to 1800 two times. But if you look at the extremes of the populations, then you have almost a 15-fold difference in cost between the predominantly African individuals and the predominantly native ancestry individuals. I'm going now to move to another example of polymorphisms and association with drugs, according to the CNPq guidelines for the thiopurines. And I will start by showing you, the major pathways of the pharmacogenetics of thiopurine drugs, which are used mainly in the treatment of leukemias, particularly lymphoblastic leukemias, acute lymphoblastic leukemias in children. So the mercaptopurine, as a standard thiopurine, has three major metabolic routes, and the one that leads to the active compound, to the active metabolites, is this route that involves several enzymes. And mercaptopurines are then, of course, a prodrug. That is, it needs to be activated in the body to generate active metabolites. One of these enzymes that leads to an inactive metabolite is the thiopurine methyltransferase. And this enzyme is encoded by the TPMT gene, which is polymorphism, but very polymorphic. This is one of the bases of pharmacogenetics, one of the first examples of the importance, the clinical relevance of pharmacogenetics. And there are well-known, well-recognized polymorphisms in the TPMT gene that are null, which result then in loss of activity of the enzyme. When this happens, you have a deviation of the metabolism to the other routes, which lead to an increase in the active metabolites and can lead to toxicity. And it has been shown many, many years ago that this has a very significant clinical relevance, such that individuals that are homozygous for these null variants in TPMT are highly susceptible to intolerance, to toxicity. Heterozygous are also at a higher risk than those that are homozygous for the wild-type allele, that do not carry the variant no-function polymorphisms. So, in more recent years, starting in 2016, it has been clearly shown that polymorphisms in another gene, the NUD15 gene, are also important for intolerance to thiopurines. And this study carried out in 3 different countries shown that the tolerance to thiopurines, the dose that the children can tolerate, very largely depending on polymorphisms in the NUD15 gene, such that those that carry in homozygosis variant alleles, the 3 allele, are highly intolerant in all populations. The heterozygous are less tolerant than the homozygous, showing clearly there is a gene dose association effect of the NUD15 polymorphism, particularly one polymorphism, which I will show in the next slides, and the tolerance to thiopurines. So here is what happens if you have the NUD15 is the enzyme that converts the active back into inactive metabolites. And if you now have polymorphisms in this gene, you also lead to accumulation of the active forms and leading to toxicity. So that the guidelines for thiopurines were reviewed in 2018, and they are now based in both genes, not only on TPMT, as happened in the previous, the 2 previous guidelines from 2011 and 2013. And we now adjust with the recommendations for the recommendations from the guidelines are to adjust the thiopurine doses based on the on polymorphisms on both TPNT and NUD15. But this is the NUD15 is interesting because the polymorphism, the major polymorphism that leads to intolerance, is absent in the European and the African cohorts of the 1,000 genomes, and occurs at the highest frequency in East Asians, in the East Asian cohort of the 1,000 Genomes Project. When you look at the admixed American populations, the frequency is 4%. And since we know that the Latin American, admixed American populations have as major ancestral roots Africans, Europeans, and native, then since this polymorphism is absent in African and Europeans, it must come from the native ancestry of the Latin American populations. And what we see is that in the four groups that are comprised in the superpopulation, the frequency of the polymorphisms increase from Puerto Ricans to Colombians to Mexicans to Peruvians, and this increase goes along with the increase in proportion of native ancestry. So that's one more indication that probably the presence of these polymorphisms in Latin American population is associated with native ancestry. And we know that, or we assume that the peopling of Latin America by native, the native population of Latin America originates from migrations from Asia, whether through the Bering Land Bridge or the coastal line. And so there is an alternative hypothesis that the peopling started from Europe, but I think the most specialists believe that these are the two most relevant routes, such that it's not surprising, maybe, that the presence of the polymorphism in the Latin American populations is associated with their Asian ancestry through their Native American ancestry. And so we decided to look, well, then what is the frequency of these polymorphisms in native populations living in reservation indigenous areas in Brazil? And we studied four groups. And what we observed was that the frequency in the Caingan group from the south region of Brazil is the largest, this group has the largest frequency of this polymorphism reported so far for any population in the world. 31% compared, as you might remember, 10% in the Asian population in the 1,000 Genome Project. The other group of Caingan is in another reservation area, 26%. Guarani in the central west of Brazil, in the southeast of Brazil is 19%, and in Chavante, in the central west region of Brazil, was 5%. So what we see is a frequency, and just to tell you, in the non-indigenous Brazilian population, whether self-reported, white, brown, or black, the frequency is less than 2%. So what we have here is a large variation in frequency of this polymorphism among individual indigenous populations living in reservation areas that shows a very high genetic divergence assessed by the fixation index statistics. And again, this is the largest frequency of this polymorphism ever reported. So next, we look in the Amazon region. And here we are not dealing with people living in reservation areas, but people living in rural areas, but not reservation areas, where the admixture is extensive, as shown here. The native African and European populations of about 130 individuals. And you see that those that have more than 80% of native ancestry estimated with ancestry informative markers have frequency, minor allele frequency of these NUT polymorphisms of 6.7%. As the native ancestry reduces, it goes to 5%. And in those with largely predominant European ancestry, it is 2.2%, which is approximately what you see in Brazilians in general. So I'll go from this study in the NUT 15 to the last slides of this presentation, where I would like to show you the effort we have been doing at the Brazilian National Cancer Institute to implement a pharmacogenetic testing program. We started that just before the pandemic in 2019. We started by creating a task force involving various units from the institution. We selected which target gene pairs would be, and this selection was taken based on the drug gene pairs represented in the CNPq and in the Dutch pharmacogenetic working group guidelines, and is focused on these 3 drugs and these genes. We approved the project by the institution ethics committee. We developed and implemented several standard operation procedures, and importantly, we created very clear procedures for the recommendations, very clear and very succinct recommendations for those adjustments according to the guidelines. And it's important to have this small recommendations so that the clinicians cannot say they don't have time to read it. This is an important point. So we developed procedures to communicate the results to the attending physicians, and we included the results of the analysis into the electronic medical records at our institution. And this effort was first published in 2020 in the International Journal, and also for the Brazilian oncologists, we made a smaller version of it in the Brazilian Journal of Oncology, that is the official organ of the Brazilian Society of Clinical Oncology. So the results obtained up to 2 months ago for the thiopurine drugs in children with acute lymphoblastic leukemia are shown here. We have so far, until 2 months ago, genotype 593 children with lymphoblastic leukemia, acute lymphoblastic leukemia, and from 9 institutions. So this is sort of a national project. We have we receive patients. We don't receive them. We receive the DNA from patients from 4 different regions of Brazil, and we genotype these patients and provide the results to the attending physicians. And here are the results of the metabolic phenotypes inferred from the diplotypes, and the dosing recommendations are based on the compound phenotypes, whether the children are normal metabolizers, intermediate metabolizers, poor metabolizers, or compound intermediate metabolizers, that is, intermediate metabolizers for both enzymes. And this is our report that is very succinct. That is what goes into the electronic medical record. It's in Portuguese, of course. So this reported pharmacogenetic test for thiopurines, the patient, the tests, the genotypes observed, the diplotypes inferred, the metabolic phenotypes, and the recommendation of the initial dose of thiopurine, start treatment with the usual dose of thiopurine, and adjust doses, the subsequent doses, according to the extent of myelotoxicity. And those adjustments should be done at intervals of 2 to 4 weeks, which is the time required to attain steady state for the pharmacogenetics of thiopurines. And then we provide additional information on how the tests are done, what is the rationale for the recommendations, in providing a link to access this information. So this is a child that had no polymorphism, so the recommendation to start with the usual dose. And here is another child that has polymorphisms in two loci in different genes, in NUDT and in TPMT. The inferred metabolic phenotypes are slow for TPMT and intermediate for NUDT. So this is a child with very high risk for toxicity of thiopurines. And then we have the recommendation to drastically reduce the initial dose and then adjust the dose according to the extent of myotoxicity. And the rest is the same of what I read for you before. So this is my last slide. This is the results obtained for irinotecan and fluoropyrimidines that we test for the for the UGT1 gene and the DPID gene. And we use 1 polymorphism here and 4 polymorphism here. And based on the genotypes, we then make dosing recommendations for patients that are candidate to these drugs, which are mainly patients with gastrointestinal cancer. So I think that my 40 minutes are over, so I'll skip this slide. And thank you for your attention. And I would be very pleased to answer your questions. And if you need any additional information, this is my email for contact. Thank you very much. And these are the agencies that finance our work. Thank you.
Thank you so much, Professor Cortez. A very, very nice presentation, huge amount of work. I really enjoyed the presentation, and I think also the attendees enjoyed it. Please, if you have any question, please write it in the chat, and I will start to read it. So I have a question here, which is how much self-declared race ethnicity correspond with genetically determined ancestry?
Well, you saw that slide that I showed, one of them that shows both self-declared white and self-declared black individuals. And you see that you can have for both groups and also for the brown individuals, what in Portuguese we call them pardos, that's the official term. There is a very, very large variation in individual proportions within each category. So someone who says, oh, I self-declares white may have 70% African ancestry, but still phenotypically finds that he looks white, that he should qualify, should declare that he's white. So it's very large. The correlation is one of my colleagues who is a population geneticist uses the expression, the correlation between skin color with self-declared is the major factor for self-declared color. Skin color and ancestry in Brazil is tenuous at best. And I think that expresses it very clearly. But on average, individuals who self-declare to be white have on average larger European ancestry than individuals that self-declare black. And the opposite is true. So on average, yes, it works, but individually, not. And I think that applies throughout Latin America, and that's the major point in understanding admixture, is that average is just average. And average is not a parameter for individual medicine, personalized medicine.
Thank you so much for answering the question. So I have another question here. Did you consider environment in modifying the effects specific loci between the Mozambican and the Brazilian black population?
Sorry, if I consider what the environment? Yes?
The environment, yes, the environment.
Well, I am sure, you know, when I talk to students, when I introduce pharmacogenetics to students, I show a pie with several slices. And one of them is genetics, one of them is other medications, another one is adherence to treatment, another one is diseases, another one is smoking, another one is drinking. So when pharmacogenetics, the slice of pharmacogenetics, is the same size than the other ones, pharmacogenetics is irrelevant clinically. You can show an association and publish a paper, but it's irrelevant clinically. So pharmacogenetics becomes relevant when the genetic component is predominant or is determinant of the drug effect. So, of course, the environment in Mozambique would be very different from the environment in Portugal. And actually, I was in Mozambique to collaborate with my colleague Albertino, and was a very, very interesting, very, very nice experience. I enjoyed it very much. And I had a very positive experience with teaching a course in pharmacogenetics that was a very attentive and very interested class. So environments will be different, and it will affect. So maybe for some polymorphisms, the environment will be more important than for some drug gene pairs, environment will be more important than the genes. And yes, this can never be, you cannot analyze pharmacogenetics without taking into consideration environment and other variables. I mean, diseases, and habits, and other aspects, many of them.
Prof Nagy, are you still there?
I wonder if you froze.
I think you might have been cut off. But yes, I really would want possibly to ask if there's one person who's got a burning question. I'm just stepping in because I can't hear Professor Nagy. Anyone with a burning question, you can open your mic and ask. Yes.
Yeah, thank you very much. Very, very nice talk. Thank you very much. I really appreciate the details. So this is more fundamental question. We believe there is only one race, human race, but at the same time we try to divide people by genetic markers. So is there any paradox why, especially in Africa, we consider just one ancestry, Black ancestry? We know that if you go East Africa, West Africa, or North Africa, it's a completely different ancestry. So are we doing the right science in just, assigning ancestry for Africa, which is, the source of everything, just putting one ancestry and then, looking pharmacogenomics to be one box or the other? I was wondering your thought about even the term ancestry given for Africa population.
Yeah, that's a very important point, and you see it applies also, and I don't know if it applies more, but it applies extensively to the native ancestry too. You see, I will come back to Africa in a second, but in one of the slides I showed you, the one on the frequency of the NUD15 polymorphism in the different native groups genotyped in our lab. So theoretically, if there are genotyping errors, they are the same. We saw a very large difference for that one polymorphism. In one population was 5% in Chavante, and in the other population was 32%. So that's a six-fold difference among indigenous populations. So, and when we use markers for the indigenous populations, or in the past, when you use a panel, we take indigenous populations, I'm sure that are not representing the extent of the diversity among native people. And I'm sure that the markers for Africans also do not express or do not represent the variety, the diversity of the within African populations, which of course I'm aware that they are present. And that's why when you do genome-wide approaches to separate ancestries, this problem is reduced. When you use a set of markers, it's one thing because you have to select the markers from a given population. But if you do genome-wide studies, particularly some of the studies we have done that we use markers from the 1,000 genomes overall population, at least we are covering the various African populations that are included in the 1,000 genomes. So, but that's a very important point. And I'm sure that if you use different markers for different panels, you come out with different proportions. There's no doubt about that. Yeah. So when you do comparative studies within your cohorts, then when you do comparative studies is fine. When you're going to do studies comparing your cohort that was done with one set of markers with another cohort, analyzed another set of markers, and then you are in trouble.
And just to add to that question, ultimately, when we look at pharmacogenetics, we are trying to go towards precision medicine, where as much as those biomarkers we are looking at, let's say, different ancestry populations, we want to understand what is happening in an individual before we give them a drug, ultimately. So, because one might call themselves African, but when you look at the genetics that is going to actually be the baseline for the drug response, it might not be African as much as they feel as an African. So ultimately, as much as you feel like we might be dividing people, but the ultimate end game is to treat each patient according to their underlying genetics, not as an average, like what Guilherme said, but as that individual with their individual genetic makeup. Thank you.
Yes, I fully agree. I fully agree with that. Actually, I try to, the reason that we select those, that we devise those 3 sub-cohorts is to show that look, when you look at the average, you don't see anything. But when you say, okay, let's take the individuals at the extreme, because there will be in the population individuals, Brazilians who have 90% African ancestry. There are Brazilians with 90% native ancestry, and they are not represented in the average, because in the average, the Brazilian population, the average African ancestry in the Brazilian population is estimated to be between 30 and 40%. And they are not represented there. So this point is very critical for precision medicine. Average is not a parameter, but for the people who are deciding whether they're going to finance that, whether private institutions, which is not our case, or the public health system, which is the situation of our institute, when you present the proposal to create a pharmacogenetic test, you have to say, look, we are going to find enough people that justify to do the tests. So it's very critical, I think, in our countries, both in Latin America and in Africa, it's very critical to do cost-effectiveness analysis to implement pharmacogenetics, because otherwise we don't know if it's worth doing it.
Guilherme, there's another question in the chat where someone is asking whether it is possible to use pharmacogenomic biomarkers for epigenomics, like in Alzheimer's disease or diabetes.
I am not aware of that. I really have no information. I was interested in epigenetics, in pharmacogenetics a few years ago, and but after that I haven't followed it. But I really can't answer that. I don't know.
We are at the top of the hour. We are at the top of the hour, so I would want to take this opportunity. Can you still hear me?
I can.
Yes.
Yes, I would want to take this opportunity really to thank Guilherme for presenting in this forum. As Africa, you can see that we are very enthusiastic to to also be part of the of making history in understanding our populations. You did take us through quite a lot of work that we have done, which has included looking at ancestry, ADMET genes. You also reminded us that we must be careful when you look at p-values. We must be mindful of the magnitudes of effects. You also then you showed us that we must also be mindful of the NNG, the number needed to genotype for us to get certain things, and very impressive work that you have done on the thiopurines with the NUDIT15. I think we would really want to look at some of these genes in African populations. They've been very quite understudied. And then you also touched on the peopling of the Americas, looking at the movement from Asia through Beringia and the coastal areas. I think in Africa also we have heard many times our history being told. It's almost like the history of the lion being told by the hunter. So we really would want to really understand also the movement that has happened within Africa itself, for us to understand some of the genetics that we have. So I really would want to thank you, and thank you for sharing your number. I'm sure a lot of people will contact you within for advice. And just to comment that Colin Masimirembwa was also listening. He is in another meeting, but you have got a long history. You did write a book on pharmacogenomics earlier on, and he wrote a chapter in that. And Colin is he is the current president of the African Pharmacogenomics Network, and we really want I really want to thank you on behalf of the organizing committee, and to thank Professor Mohamed Nagy for moderating this session. And also lastly, just to thank the audience for being with us here. Thank you so much for staying the course. We hope to continue giving you exciting seminars, and we are moving into education as well. We will come to you, Professor Guilherme, when we need some support in different areas. And before I hand over to Vicky, I would want to just you to say one word. You are coordinating all this. I really would want to give you an opportunity to close this session. Thank you. Vicky, over to you.
Thank you, Prof. Just to let everyone know, and thanks to Professor Kurtz, this is really an informative session. Just to let everyone know that the webinars will be continuing until at least March next year. And from March, as Professor Dandara mentioned, we'll have a proper training for researchers who want to conduct pharmacogenetics or pharmacogenomics related research. So thank you. We look forward to meeting again next month. We'll send the details through before the end of this week of all the other speakers. Thank you.
Thank you.
Thank you. Bye-bye.
Bye. Thank you.
Bye-bye.